Zoom, and Zoom I can do everything. So I'm so sorry about this technical problem. First of all, I will present a little bit myself. So my name is Youssfi Abdessamad. I did my PhD in NCS, the school at University Mohammed Zag, Rabat, in 2021. After that, I did my first postdoc or my postdoc at Maser Foundation for Research Science. So for my PhD, it was more especially for monitoring videos and deep learning model and statistical model. It's more especially for developing or have an like as a outcome of my PhD system, a monitoring system to monitor the elderly people in their own house or in the hospital or something like that, so, or where they stay generally. So after that, during my postdoc, I developed a model, a deep learning model for the satellite images, or more generally for remote sensing. So the goal or the main idea of this model is to detect the farms in an agricultural area, and after that, like the results of this part will be the input of the second part, which aim to like to have a prediction of the production of like the tree or anything or in the agricultural area. And we have a good results for this model, which comes in two parts. Now I'm working as a research science at University of Miami. I am working more especially for medicine data and working for the genomic data, including the RNA-seq data and the whole exome sequencing or the whole genome sequencing. We will have an idea about what is RNA-seq data and what's the whole exome sequencing, what is the whole genome sequencing. So today I will present a little bit what is like I do now, and what is the pipeline that I follow to analyze this type of data, and some example of using also deep learning using images to predict and or to help the prognostic or and the diagnostic for physician in the hospital, like to make a decision about cancers, more especially for cancers. So generally, can you go to the next slide? So generally, during this presentation, I will like let you have an idea about what is the data packaging or the data processing. For example, we have for genomic data, we have like two parts. The first part is done by the biologic staff using machines, specific machines to have like sequencing. We have like, for example, some patients or some samples taken from some patients, and we extract like the RNA-seq data and the whole exome sequencing and the whole genome sequencing from those samples, in order to have at least the RNA-seq data and the single nucleotide variation and structural variance data, and also the copy number variation. So after that, for today, I will just focus more for the differential expression analysis to extract up and down regulated genes, which based on using more especially the RNA-seq data. So like the biologic staff have like their own they have their own like pipeline to follow or to progress or to do in order to extract the RNA-seq data. After that, we take this data and we apply some analysis, we apply some algorithms in order to extract like some information from this data. So for this presentation, I will focus more for the RNA-seq data. After that, of course, yeah, we can follow the it's the same process or the same pipeline that we have to follow, but we have like to adapt this pipeline or this steps based on our strategy. What is the main idea? What is the story about our research or about our analysis? What we mean like to found after applying those algorithms or those methods or this analysis. So we have to define the idea or the strategy that we have to follow in order to have like more information about what we mean to found. And after that, we have to check those genes. For example, I have the RNA-seq data. I extract some genes, but this list of genes which are significant for me after the analysis doesn't mean anything for the biologic stuff, or it's mean something for the biologic stuff. So we have to check like the pathway, what is the mean of those genes, what's those genes like do for the biologic stuff. And after that, I will just show some example of using deep learning model, more especially the convolutional neural network model, to have like a prediction, prognostic, or diagnostic for cancers. Can you go to the next slide? So for the steps, there are some steps that we have to follow. So we have the experimental design. We have like some mouse or some samples extracted from some patients from a hospital or something like that. And we have like some stuff from the experiment. So we have to extract the sequencing or to do the sequencing RNA from these samples or from the mouse that we have in our laboratory, or we have in the or the experiments done, for example, by the biologic stuff. For example, we can apply the vivo or the vitro experiments. We can apply also the PCR. We can apply a lot of like model or methods that we can apply in order to extract like the RNA sequencing. After that, when we extract the RNA sequencing, we put them or we save them inside some files, which are the BAM files or SAM files. The BAM files or SAM files, they are like a binary files which contains the sequencing of the RNA extracted from the sample that we have in the laboratory. So we have to apply site alignment using those files in order to extract. For example, if we have just RNA-seq BAM files, we can extract the RNA-seq data. If we have like the whole exome or the whole genome sequencing, we can extract the structural variance data. We can extract the copy number variance data, and we can also extract the single nucleotide variant data. And after that, for example, for the RNA-seq data, I'm focusing more with the RNA-seq data. We have to apply some normalization. Why the normalization? The normalization, just because we have like the genes and we have the samples. The data is like a matrix, which like for each genes, we have like the counts for the sample. So the count is very big, is very high. So we have to normalize this data in order to have like more concentrated data. 1, 2, in order also to have like an output which can be visualized and which can be analyzed by like physician, by biologic staff, by someone who doesn't like understand very well the bioinformatics stuff. And after that, we have like some hypotheses. We at the beginning of or at the start of our analysis, we put some hypotheses based on like the outcome or the output of the biologic stuff. So we have to test those hypotheses. For example, we can take a gene, for example, as an example, gene MEK. So MEK is a gene which is very famous, for example, in myeloma cancer or lymphoma cancer. And for example, in our study, we have also to check like if what we found after our analysis is aligned with what the bioinformatics staff found like during their experiments. So we have to test the hypothesis, and we have to check if we arrive to align our results with like the results which the bioinformatics staff found. After that, we have to do the downstream analysis and genetic analysis. Genetic analysis, it's like using the structural variance data, the copy number data, variance data, in order like to just to check more and more and more the importance of these genes. For example, you can check the copy number variance in order to find if like the type of mutation, there are a deletion, there are a duplication, a translocation, or something like that in this gene or close to these genes which impact the cancer. So we have to do like the genomic analysis also. Can you go to the next slide? I'm so sorry. So for the next slide here, we have three parts of analysis. The first part, it's the library preparation. So we have the total RNA, and we have like to select the mRNA, and after that we have to make some directional adapter ligature and reverse the transcription. Why this step is so important? Because after that we will like apply some methods to have the sequencing of the RNA. So we have to adapt like our data or our issue in order to have like a very good sequencing after applying the PCR amplification or the barcode addition. This step is done. It's more specially for the biologic stuff, and it's more specially done by some machines in some laboratory. And after that, we will have, after this part, we will have like the BAM files. We can take those BAM files and we can apply some short read alignments. So for the short read alignments, it's depend to the type of the data that you have. If you have the RNA-seq data, you can, for example, convert. There are two like type of coding of the sequencing or the BAM. It's HG19 or HDG38. So just to follow like the stage of art, we have to have the files in HG38. It's just a type of coding of those files, nothing else. So if we have like HG19, we have to convert those BAM files to HG19, and we have also to align those files using a package which is called STAR. This package help us to more extract the data from a sequence, because the sequencing is just a sequence of HACG, but from the sequence of those like the alphabet, we have to extract like a matrix. We have to extract some values. So for this reason, we have to do the short read alignment. For the short read alignment, as I said, it's depends on the type of the data. For example, if you have the whole exome sequencing data, it's another step. If you have the whole genome sequencing data, it's another step. There are approaches which called MGP 1000, which is very famous for the bioinformatics or for the genomics, like specific specialist, which is very used to align the BAM files. And after that, we extract like data. We extract Excel files, which are a matrix of lots of informations. For example, for the RNA-seq data, the count of the genes for each sample. For example, for the copy number, it's the total copy number, the minor and the major copy number for each position and or each breakpoints. And for the SNV, it's like for each breakpoints we have like some informations that help us to extract like information from this data. For the whole for the RNA-seq data, we have to apply the normalization, as I said, like in the previous slide. And after that, we apply differential expression analysis, and we check the gene ontology analysis. And after that, we can, for example, have an allele specific expression, genetic analysis, or bioinformatics like analysis or models that we can apply based on the results of gene ontology analysis. For the bioinformatics, for example, when we found like some genes which are significant for our analysis, we have to check like the copy number, to check the type of mutation, to check what happened exactly in this position or in the position of genes. And like it's not in all the patients. It's impossible that we have like the same information for all the patients, but for some specific patients, we can found like something very specific that can help us for the analysis or for the whole analysis or for our whole analysis that we are applying or we aim to do using the RNA-seq data. Can you go please to the next slide? Yeah. So for the next slide, here we have some packages that we can apply. For example, for short read and alignment, we can use star. Star, it's the power used like package. It's the best one for the alignment. We can found also HI SAT2. For the read count, we can use HTSEQ or some tools. For me, for example, for the short alignments, I use stars, and for the read counts, for me, I use some tools or BAM tools. There are also another package which is called BAM tools. For example, if you remember, as the stars, I said like for after the alignments or after the sequencing, we can have files as a BAM files or as a SAM files. For example, if we have a BAM files, we have to apply BAM tools. If we have a SAM files, we can apply SAM tools. So what is the main of using these packages? This package is aimed just to convert the coding of those files. We can also apply those packages in order to convert the SAM files to FASTQ files. So for the FASTQ files, generally it's not always, but generally we have to read, like we have for each BAM files, we have to read the first read and the second read. So we have to convert them to FASTQ files, first read and second read. After that, we take the both files for the same sample, and we convert them to just one file, which take on consideration the both of read. For the differential expression analysis, there are lots of packages, not just those four packages, SEQ, the DSEQ or the XSEQ or HR or VOMLIMA packages. Can you go to the next slide? So for the data normalization, we have SVA, SEQ, and comma or peer or SNM. So for the normalization, we will see more specifically what is mean normalization or why the normalization. As I said, the normalization help us to more analyze the data and more also have significance data. If we take just the counts like as the first output of after the alignments, we will have some results, but it's so hard to read those results and it's so hard to visualize those results. So for this reason, we applied the normalization. Can you go please to the next slide? So for the alignment, as I said, after the sequencing, we'll have like, as the figure show here, or as shown here in this figure, we'll have like a sequence of A, T, and C and G. So from those sequencing, we have to extract a data as shown in figure C. So for this reason, or this is the main idea of the alignments. We have the exome, or we have the genome, or we have the RNA, and after the sequencing, we will have like a sequence of alphabets, and from this sequence we have to extract a data which contains values, which contains like some information that we can based on our analysis. Can you go to the next slide? So after the alignments, we will have for the RNA-seq data, for example, an Excel file. If you can see, for example, here for this heat map, the expression heat map, an Excel file for the columns, it's the samples, and for the rows, it's genes. So for each genes, we will have like a count for each sample. And after that, we have to apply some analysis using this matrix to visualize or to have an idea about the clustering, or if we have like two clustered group or not, or our data, it's so significant. It's not significant, but it's can help us to find very fast, or to go so fast for our analysis. We can apply the PCA and we can visualize the PCA. So the PCA help us just to find more clustered genes or more clustered samples, and those samples like have the more clustered samples have like comments. How can I say? Comment like futures or comment values, or it's more comment futures, and like if sometimes we can't find like a two clustered sets or two clustered group. In this case, it's not hard, but it's probably can like have after the analysis, we can't have like a very significant results or a very significant plots after our analysis. Can you go to the next tape? Next slide, sorry. So for the normalization, there are many like methods that we can apply. We can apply the CPM, which is the count per million. So for the CPM, it's aimed to adjust for differences in sequencing depth, cross like sample, and provide relative experience value, and compare scale by scaling the raw read counts and each gene by some samples specific genes, like the total counts, and multiplying by a scaling factor of 1 million to obtain the count per million. So for the CPM, we take like 10 times a million times the read count mapped by each gene. Go to the next slide. After that, we can for example, for the normalization, we have to use just one of those methods that which I'm trying to show now. We can also use the transcript per kilobase million. This one, it's the more like used, and it's the best one, or it's better that if we use the TPM or TMM for the normalization than using CPM or another one, or the FPKM or the RPKM. So for the TPM, it's the first normalization for gene length. So it's used for, if you remember, I said that for the transcript, we have like two reads. So for the TPM, we can use the both read to normalize the data. It's not like the CPM. And the it's making the sum of all the TPM for each samples identical. And this allows for a more accurate comparison of gene expression between samples. So for this reason, we prefer to use GPM than CPM or another method of normalization. Can you go to the next? So the next one, it's based on using the FPKM or RPKM. So the most basic one, it's the FPKM and the RPKM. It's read per kilobase, for example, the transcript per million mapped read, and it's closely related method. So the fragments per killion of transcripts per million mapped read, and those techniques normally for both the length of the gene and the total number reads, and make expression. It's help us to make the expression comparison between genes in the same sample possible. Can you go to the last one? So for the TMM or the trial mean of M value normalization, it's like used typically in or by default in each R. So we don't have to apply this method, and it's able to calculate the scaling factor based on the weighted trial mean of the log expression ratio. And this method is so robust and the high for the against the high count trees and the difference in RNA composition. So why it's more used for each R? Because like when we have some genes which have a high counts in the RNA-seq data, so those genes are so important for us. For this reason, TMM it's used by default in each R, because each R or the package each R, it's the first package with or the best package which we use for the differential expression analysis for the RNA-seq data. Can you go to the next slide? So here, for example, a sample example for the RNA, the log CPM. So for the read count, generally, it's like hundreds or thousands for the read count for each gene. But after the normalization, if you can see, for example, here we can find like 0.5, 0.25, 0.20, 0.15, 0.10, 0.05. So we can reduce the values of the counts, and like that, so we can concentrate our data to the center. This one, it's an example of the normalization. It shows us like how the normalization it's so important for our analysis for the RNA-seq data. And for this reason, like in a bioinformatics, like the first step that he can apply or that he can do for the RNA-seq data is the normalization after applying any analysis methods or any analysis process. Can you go to the next slide? For example, here we can see for the data which are not normalized and the data which are normalized. You can see here, for example, like for all the samples, because the rows is the samples and the column is the count, or yeah, it's the count. So we can see, for example, here, like the samples are very close between us, but if we use just the data like that without normalization, we can see, for example, the box plot are not close for all the samples. Can you go to the next slide? So this slide, it's about applying the PCA. So what is the main idea of applying the PCA? The main idea of applying PCA, it's just to have an idea about how our patient or our samples are close between them. So for example, here, our for the first plot in the left, we have three type of samples. We have LP, we have ML, and we have base. So for example, here we can see the samples are closed, like the base are closed between them, LP are closed between them, and ML are closed between them. And after that, for example, for the sequential line, we can do that also. For the sequential line, we can see that's the groups are not close between them. For example, if we can take L004 and L006, we have like just one outlier from this group and one outlier from the other group. But for example, for L008, we can like see that's this group is more close to the other groups. So the main idea of using the PCA, just to have an idea about if the features of the groups are like comments or more close for all patients, taking in consideration the groups or the subgroups or something like that. Can you go to the next slide? So for the next slide, here we have after the normalization. Normalization is more for the samples, but for the genes, we have an RNA-seq data. We apply the normalization and we check this data, but not all of the genes are so high expressed or so high, have a high count. For this reason, we have to remove like the gene which have like a very small read count. So what we have to do? We have to just to apply the VOM packages which aim to remove the more slow, like the VOM packages which can show us, like for example here at the right for all the genes and at the left after removing the genes, and we can see, for example, at left subfigure that the genes become more like good to analyze. So for removing the genes which are like, or which have a very small read counts, there are a lot of methods that we can apply. For example, we can check the genes which have, we can put a threshold and we can check the genes which have like for all patients or for 90 patients or for a very high percentage of patients or the samples have a very slow or a very small read counts, we can remove those genes. This one, this is the first one. The second one, we can just plot the density of like the counts, and we can like specify a threshold, like the peak, and we can take this peak as a threshold, and after that we can remove all the genes which have like read counts more small than this peak. This is the second one. The third one, it's using just the the HR the VOM-LIMA package, which based on using, it's used by default, which based on using like a threshold by default, it's 0.05. That's mean we keep all the patient or the genes which have like a threshold more high than we keep the gene which have like 95% of the samples which have a red count more high than the threshold. Can you go to the next slide? So after that, what we have to do? We have to apply a linear regression, and we have to check the prediction model, and we have to check the distribution of this linear regression with the distribution of the prediction model and the distribution of the input model. And after that, we will have like an exponential function. That's mean that our like process is going very well or is going very good. And after that, we have to check the results that we found. So for example, here, if you remember, we have three subgroups: base, LP, and ML. So we will have like two comparison. It's basal versus LP and basal versus ML. So we can use the Venn diagram just to have an idea about the number of the gene which are significant using a threshold of log4 change and p-value. The log4 change and p-value are a statistic criteria that we can based on in order to like do our analysis. It's the output of the VOM-LIMA packages. And for example, here we can use the Venn diagram in order to specify which the number of genes which have a p-value less than 0.05 and significance for us, and also for the log4 gene which is high than 1 or less than -1. So for example, for the comparison which between basal and LP, we have the sum of 864 and 2784 genes which are significant for this comparison. And same thing for base L versus ML, we have 150 and 1050 and 2784 sum of those values as a number of the genes which are significant for this comparison. And like an intersection between the two comparisons, we have 2784 genes which are significant for the both of the comparisons. And the number of genes which are not significant, that means we will not take in consideration those genes for our analysis, is 11,926. You can go to the next slide. So after the VOM-LIMA package or results, we will have an Excel file which is like this, where we have the gene, the enzyme ID, the TX chromosome, like the chromosome where we can find like these genes, and the sample are the name of genes, the log4 genes for each gene, the average expression like average count, and t value and p value and the adjusted p value, and we can find also the average for gene. Can you go to the next slide? So this one is just to check like the more significant or the 5 more significant genes we can use it. It's very useful, for example, just to have a very quick idea about the genes that we found. Can you go to the next slide? After that, this plot is called the volcano plot. So here, for example, we can have a plot just to visualize the genes which are significant for us. So for here, for example, we have the dots in black. The dots in black are the genes which are not significant for our analysis. So we will not take in consideration those genes, but the dots on red are the genes which are significant. For example, here, if you remember that for the Venn diagram plot, we take in consideration two thresholds. It's the p-value and the log4 change. It's the same thing here. We take the p-value for the y-axis, and for the x-axis we take the log4 change. So for the gene which have like a log4 change more than 1 are the genes in right, and which have like a log4 change less than -1 are the genes which are in left. So for the genes which have a log4 change higher than or more than 1, they called like the upregulated genes, and for the others, the genes they are the downregulated genes. Can you go to the next slide? So as a visualization, we can use the heat map. The heat map, it's very, very useful to have an idea about our analysis, it's good or not. So for example, here, if we can go back to the hypothesis that we put, we have 3 group of samples. So we have base L, we have LM, and we have LP. We can see here, for example, from the heat map that for the base L, for the genes, upregulated genes, we can see the genes in blue, and same thing for the in up and down, and in red it's the genes which are downregulated. But it's the opposite for the others, for the others like genes, the others group LM and LP. So the heat map, it's very, very useful to visualize your data and give us an idea about our analysis is good or not. And it's also give us an idea about the genes and the importance of genes, of those genes for each group of samples that we have. Can you go to the next slide? So after that, we have the list of genes which are significant, the up and down regulated genes, and we have to check using the read count data, just taking in consideration the significant genes. We have to check like the pathway or the oncology enrichments of those genes. So for this reason, we check the pathway. For example, here we can like have some pathway which are so significant. For example, DNA replication, chromosome segregation, DNA templates, DNA replication, mitotic cyst chromatid segregations, cyst chromatid segregation, etc. And each pathway, like we can visualize each pathway using the count. This plot, it's so significant for the biologic stuff, and it gave them like an idea about the analysis that we apply using like the RNA-seq data, and give them like some information about the meaning or the enrichment oncology of the genes that we take on consideration like as a significant genes. Can you go to the next slide? So it's the same thing here. We can visualize also the results of the pathway, yeah, taking into consideration the number or the position in the ranked list of genes, and also the running enrichment score. So the running enrichment score is very, very famous in bioinformatics. To give us an idea about like the position of the list of genes and how those genes are so significant for the bioinformatics stuff. Can you go back to, can you go to the next slide? So here we can just visualize the list of pathway that we found so significant for us as a table, and after that we can like report this list of pathway for the bioinformatics stuff. And for the bioinformatics stuff, those information is so important, just to have an idea if the analysis that we did is so much with the experiments done in like the laboratory, for example the vivo or the vitro experiment or the screen method experiments. Can you go to the okay, so now I will just give an example of using, for example, because in bioinformatics there are two parts. The first is passed on using like the RNA-seq data, the whole exome data, the genomic data generally, and the second part is based on using images. For example, here it's just an example that we can apply, for example, for lymphoma. This paper is used for lymphoma. We have like a sequence of images from like a patient or a sample, and we can have like a prediction model using this sequence of images, and we can use like as a first step it's not autoencoder, but just the first parts, just like to extract the information from those images. And after that we can use like a sequencing model using LSTM, JU, or etc. For example, here they use attention network just in order to have like after that a probability of the prognostic or the diagnostic of these patients based on images taken from these patients. Can you go to the next slide? So it's the same thing here, but here it's for another cancer. It's for the breast cancer. For example, here we have a images, and we can just apply a sample deep learning model. This one is more just for classification of the lymph nodes, and because for the lymph nodes, there are a lot of like lymph nodes, and we can just apply a CNN model to classify the lymph nodes. Can you go to the next slide? So this model, it's more especially for the prognostic. So here, for example, we have genomic data, and we can extract like from the genomic data, or we can build a graph from the genomic data to extract like the information. And after that, we have like we can apply a pooling, and the pooling based on the graph that's we create from the genomic data. And we can apply a fully connected layer. It's like just a Métier perceptron layer, and after that we can have like as a output from the genomic data, and we can combine the results of this part with the using images which shown in the figure B. For example, we can use an autoencoder like unit or one of the extension X units or attention units or RNA-seq units, or there are lots of extension of or trans units of units. to have like a prediction applying the autoencoder and using images. So this paper is used for the lymphoma cancer, and it show very good results. Oh, it's very useful method for like for the physician or for the doctors to have like an idea about the prediction model for the lymphoma cancer. Can you go to the next slide? So this one is for the prediction of the diagnostic. So for example, for the prediction of the diagnostic, we have an image which content like for a patient or for a sample, and we can apply a segmentation in order to have a patch generation of images or small images from the very big images. And after that, we can apply like a normalization from the images, and we can detect the blurry images from the normalized images. And after that, we can apply a linear model. It's not a linear model, but more it's a multi-perceptron linear model to train our model. And after that, we can apply model evaluation using like the cross validation or using the one lifts out or p lifts out for the validation of our model. And after that, we can visualize our result. Can you go to the last slide? So this workshop or this presentation is more especially, for example, for the impacts of artificial intelligence or the impacts of using, it's more specifically for using machine learning and deep learning for medicines or for using the genomic data. So we see the goal of, during this presentation, the goal of the differential analysis to extract the significant genes. And after that, after finding significant genes, we can check if like the results that we found after our analysis are aligned with what like the biologistic stuff puts as an input or as a hypothesis from the beginning of our analysis. After that, we can test the pathway and check the biological meaning of the results found. When we see the results found, I mean the genes which we found are so significant for our analysis. And like as a last part of the presentation, we see that also for the deep learning, there are a lot of models or a lot of papers which aim to have or to make a prediction for us or for diagnostic and for prognostic for the cancers, taking into consideration like lymphoma cancers, myeloma, breast cancers, any type of cancers. As can you go to like the last thing? Thank you so much for to assist my presentation, and of course, if you have any question, feel free to ask. I'm here to answer your questions.
Merci beaucoup, professeur ALAOUI, pour cette pertinente présentation qui est très riche en termes d'informations. Et si vous permettez, professeur, donc on va ouvrir une parenthèse pour prendre quelques questions des participants.
Oui, bien sûr.
Merci.
Merci à vous.
Donc, s'il vous plaît, si vous avez des questions, donc vous pouvez lever la main.
Je sais très bien que y a
oui.
Parmi vous si vous me permettez.
Oui.
Je sais très bien que parmi vous, il y a plusieurs personnes qui ne sont pas des médecins ou bien ne sont pas des
informaticiens aussi.
Non, ce n'est pas des informaticiens, mais...
Informaticiens et des médecins aussi. Donc,
oui, la présentation, c'est plus pour les médecins, mais
oui.
Vous pouvez poser n'importe quelle question. Donc,
D'accord, ça marche.
N'hésitez pas.
Donc, à vous la parole, chers participants.
Bon, il y a une question au niveau du chat. Je veux savoir le nom de logiciel que vous avez utilisé pour avoir les diagrammes que vous avez partagés, par exemple, au diapo 14. Alors, le diapo 14.
Je vais revenir au diapo 14.
Oui, si vous voulez. Bon, j'ai la présentation ici.
Oui.
Bon, ce sont des diagrammes qui sont réalisés par le code. Vous pouvez les faire, en fait, en Python, en R, en Java, en n'importe quel. Donc, ça dépend de vous. Par exemple, pour moi, j'ai utilisé R et Python, mais vous pouvez utiliser en fait n'importe quel package pour n'importe quel langage pour visualiser. Ce sont des plots qui sont tout simplement réalisés par le code. C'est pas un logiciel.
D'accord, merci pour votre réponse. Une autre question?
Hello.
Hello?
Hello, Professor. I was pleased to know what do you mean by image normalization?
Okay. Can you? I understand your question.
It's Medicine Diagnostic.
You mean for the slide 28, right?
Yes, maybe.
Yeah. Can you go? I'm so sorry.
Oui. Monsieur,
Madame Zara, slide 28, slide 28.
oui.
Okay, so for image normalization, it's just like, for example, you can take the images, okay? You did sliding of the images. We have a sequence of images. Like the first image, it's depend to like the first time of the patient take like a diagnostic. The second image, it's for the second time like this patient take a diagnostic, etc. Okay, and we have a sequence of images. We take those images for like, for example, the first batch for each time. We take this list of images and we apply a normalization. Normalization, you can, for example, compute the mean of the images and reduce the mean of all these images from the each image, or you can apply like if here we have the same image, the same units of measuring, so it's we can apply like the mean divided by the standard deviation. It's a normalization. It's a simple normalization.
Okay, thank you.
You're welcome. Normally, it's a good question. I'm so sorry who asked. It's Madam Nasima, right? So
Yes.
the meaning, the normalization generally, for example, I know that's a lot of participants here use deep learning model. Generally, when we download the data from our websites, it's normalized or already, okay? But
Yes.
if we use our own data, we have to normalize this data. If else, we will have like some bad results. For this reason, there are lots of like PhD students expend a lot of time of developing a deep learning model and applying this deep learning model, and they found like the accuracy is so bad, the F score is so bad. Why? Because
They
your
skip
data,
this.
yeah, because your data at the beginning it's not normalized. It's not like you have to do some pre-processing before applying your deep learning. Yeah, it's a good
Okay,
question.
I understand.
Thank you.
Yes,
Thank you.
thank you. Thank you so much, Professor.
Yeah, thank you.
Merci, professeur. Donc, vous avez d'autres questions?
I think there are a question in chats.
Okay.
Where we can find data, especially about the human resource. Okay. So the data, it's so hard to find the data. One, more especially for the genomic data. Two, it's a very big data. It's about like tens of terabytes. So you can't run this type of data in your computer. So you need like, for example, here I'm working in a very big lab. We have all the stuff to do this, but for like just for you, it's very hard to like to have a very big data. But you can find a very small data in for example, as data used from some papers, from some website, hospital websites or laboratory websites, and that's it. But for the data, like a very good data, it's very hard that you can find those type of data, or you have to apply and you have to follow a very long process, because it's a very sensitive data. It's not given to anyone just like that. Okay, you have like to have an affiliation, and you have to apply using the affiliation, and you have you understand what I mean? Yeah, thank you. Just answer the question.
Nassima, vous avez une autre question? Ou bien vous avez laissé la main?
No, I'm
D'accord.
sorry, I just let.
D'accord, pas de souci. Monsieur Mbark, merci de couper le micro. Vous n'avez pas de questions? Donc, si vous n'avez pas de questions, donc on peut clôturer.